that's not. All right, well, the projector, I think, finally died. So we'll be stuck with just the PowerPoint today. All right. That means I'm going to have to explain everything just with the pictures drawn on here. No drawings or anything, so I'll try to be as detailed as I can. Again, projector's not working for some reason, the written projector. Everything's plugged in. I don't know why it's not working. All right. So, where we last left off was something called restriction fragment length polymorphism, RFLP, which basically is the following trait. Each person has about 99.999 whatever percent shared DNA, yet we all have various different phenotypes from each other. These phenotypes arise from little tiny base pair differences in our genome that overall don't usually result in significant changes. But the times that there are changes in our genome from one person to the next, it is because of a single base replacement. You learn in genetics that they call these replacements, when they show up in phenotype differences, it shows up as dominant or recessive phenotype. In this class, we won't be asking you guys about, dominant, recessive, heterozygous, how to read what do they call these charts here? I can't remember in genetics. These inheritance charts things. You won't be asked to do that. But really, what is the most important thing is how we are able to see these traits at the genotypic level. So, over here we have a heterozygous person. I know this is a heterozygous person because they have a dominant, capital A, and a recessive, lowercase a. So they have a copy of each type of this gene here. The difference between the dominant and recessive gene here is that there's a single base pair difference right here at position 2. You don't need to know what the base pair difference is, but it's labeled right here, a single base pair difference. That single difference results in a dominant and recessive genotype here. So, visually, how we visualize this is we use enzymes called restriction enzymes or endonucleases. Back last time, we talked about exonucleases. Anyone remember how exonucleases remove nucleotides? Where do they remove the nucleotides in a DNA sequence? At the ends, right? Exonucleases remove nucleotides from either the very end or the very front of a DNA sequence. Contrast that with an endonuclease. An endonuclease removes nucleotides somewhere in the middle of a DNA sequence. So if we use restriction enzymes on the capital A genotype here, it's going to cut at the 3 labeled positions, 1, 2, and 3. It's going to cut DNA into, this example here is going to cut in at the 3 labeled positions. If we use that exact same restriction enzyme on the lowercase a gene, lowercase a genotype here, it's only going to cut at 2 places instead. The result, if we use this same restriction enzyme on both versions of this gene, the capital A or the lowercase a version, if we use it on the capital A version, we create a short fragment and a medium-sized fragment of DNA. But if we use it on the lowercase a one, we create a really long fragment. You remember back in exam 1, we have these things called, gels, gel electrophoresis, whose main job is to separate things based off of length or size, right? So, if we take this sample here, after it's been cut up into pieces, and we run it on a gel, wherever the band shows up on the gel will tell us its relative length. I will note, though, that you see this bold line right here, right? Let me zoom in a bit. You see this bold line right here in between 2 and 3 and the same one right here, right? So it turns out when you do this experiment in DNA, you have to label your fragments with some fluorescent probe. Apparently, in this example here, the fluorescent probe is only able to bind in this region of DNA. Therefore, there are no probes that can bind between 1 and 2. Because of that, we can't visualize the fragment that's going to be between 1 and 2. Anyone see? And again, let me re-explain why that is. When we run a gel with these fragments of DNA, we will not be able to see this one because there's no probe attached to it. The fragment is still made, we just won't be able to see it on the gel. So we're only able to see the medium-sized fragment, the one that's made between 2 and 3, and the long-sized fragment, the one on the lowercase a. So when we look at a gel, in this example here, here's capital A, lowercase a, here's the heterozygous person. Notice they have 2 bands because each of these bands is identifying each of these 2 fragments that are made. There isn't a third band again because there's no fluorescent label right here. So, which band is what though? You might remember in exam 1 that the way you read a gel is that anything that is further down the gel is what? Smaller or larger? If it's further down the gel, if a band shows up near the bottom of a gel, does that represent a shorter thing or a longer thing? It represents a shorter thing, okay? So if you don't remember that, write that down somewhere, is that when you're looking at a gel, whenever you have a band that is near the bottom, it is always the shorter, smaller thing, whatever it is, whether protein or DNA or RNA. If the band is closer to the bottom, it's the shorter, smaller thing. If it's closer to the top, it's the longer or larger thing, okay? Therefore, this top line right here, this top one here, must represent the really long fragment that was made. And therefore, the bottom line here must represent the medium length fragment that was made, okay? So again, notice that if a person is heterozygous, they will have both of these genes, both of these versions of the gene, the one making the long fragment and one making the intermediate fragment. That's why there's 2 bands here. Look at the person that only has the recessive gene, only has the recessive trait here, the lowercase a, lowercase a. Notice they only have 1 band, and notice it's the one on the top. That's because, and this is where I would draw it out, but I can't because the thing isn't working, they only have the lowercase a genotype. They don't have the capital A one. Because they only have the lowercase a genotype, they can only make the long fragment. All right, that's why there's only 1 band representing only the long fragment of DNA. And finally, we have capital A, capital A. That is someone that only has the capital A genotype up here. That means they can only produce the medium length fragment, which should be closer to the bottom on this gel. That's why they only have 1 band. All right, so those are the 3 possibilities that you can have here. Someone that has only the dominant genotype, someone that only has the recessive phenotype, or someone that is heterozygous and has 1 of each thing. On the exam, you will get a version of this figure. It won't be as labeled as it is on here. On the gel, it might be lane A, lane B, and lane C, and you'd be asked any number of questions about it, such as based off this diagram here, which lane represents someone who's heterozygous? Well, it's clearly someone that has 2 bands, right? It isn't always going to be that easy, though. Maybe the probes will be in different places. Maybe there will be 3 probes on the exam, or only 1 probe on the exam. Who knows? But as long as you're able to interpret what the probe position and the restriction enzyme position means over here, if you can read what it looks like over here and understand what it means on the gel, it doesn't matter what format you get on the exam. Okay? So again, contextualize, internalize this, because I've seen this on about every exam 3 for several semesters in a row. Okay, moving right along. Oh man, for this I really wish the lamp was working. Okay, let me see. Let me pull up a practice exam question. There's a very relevant practice exam question here. Let me see if it's this one. 1 second. This one. Okay. So this is on, when you go look at the practice exams, this is under exam 3, more practice, November 18, 2016 or whatever. So, we'll get to this question in a second. The next section of chapter 10b is cloning, gene cloning, and how it works, okay? We just learned about restriction enzymes. And last class we talked about that what restriction enzymes can do is that they can cut somewhere in the middle of DNA. They don't have to remove nucleotides one at a time from the end. They can just find a spot in the middle of a DNA sequence and cut it. How do they do that? They look for a specific pattern. Depending on what restriction enzyme you have, it's going to look for a specific pattern in your DNA sequence. Restriction enzyme EcoR1, for example, is going to look for this exact pattern up here. GAATTC or whatever. And every time it finds that pattern, it's going to cut at the labeled places. Cutting these exact places creates an opening in your DNA strand. And again, because this is prokaryote, it's circular DNA. It creates an opening here. Over here on the right, we have a gene we want to insert into this prokaryote. Oh yeah, I guess I should have started with the application for this. Why do we even bother doing gene editing? Well, there are certain proteins that we can actually trick prokaryotes into making for us, such as insulin. Imagine that it would be very helpful in the biomedical field if we get prokaryotes and bacteria to make insulin, a human usable insulin for us. And we do so by inserting the insulin gene into a prokaryote, and it ends up transcribing and translating it into insulin for us, and then we can use it for any biomedical application. So in order to get a prokaryote or a bacteria to make insulin for us, we have to insert an insulin gene into the prokaryotic DNA. And again, now back on track, we do that by using a restriction enzyme. So when we use a restriction enzyme on the prokaryote, it opens up the prokaryote DNA. And when we use the exact same restriction enzyme on a fragment of human DNA, it's going to cut it up like so. It's going to create open ends like this too. Notice we've created sticky ends, which are these non-overlapping ends of DNA. We've created sticky ends on the human gene over here, and sticky ends on the prokaryotic gene over there on the left. What ends up happening is because we use the exact same restriction enzyme in both cases, we've created matching puzzle pieces. We've created a puzzle piece over here on the right that will fit perfectly with the puzzle piece over here on the left. That is what this thing is. This is recombinant DNA. This is recombinant DNA technology, gene editing technology. One particular application is illustrated like this. Okay, and if you use the same restriction enzyme, they'll be able to fit together, and bacteria will be able to read this gene and synthesize it in whatever this gene happens to be. Okay, so that's the main idea here, is there's a restriction enzyme, opens up this, prepares the human DNA here, and then combines them. Okay, so let's see, that's enough information to answer this one now. This is at the very end of this practice exam here, of the exam 3 more practice exam. And you are very likely to see a version of this question on exam 3 as well. It is a very important question. So I'll read it out. A schematic of the vector blah, blah, blah is shown. The restriction enzymes listed cut only where indicated, and they do not cut anywhere else in the vector. So this thing over here, notice it's circular. That's your prokaryote. That's your bacteria thing. Okay? And here's a bunch of different places here where a bunch of different restriction enzymes cut. There's EcoR1, for example. It looks like EcoR1 cuts in one spot on this plasmid, on this bacteria DNA here. There's a bunch of other ones, though, that we've never heard of before, but that's fine. You're not required to memorize why each one is special. Just note where they cut on here. So there's the prokaryote thing. Here is the gene we actually want to insert. Let me zoom in some. Here's the gene we actually want to insert into the prokaryote. And here are the same restriction enzymes and where they cut respectively on this gene here. The question is, give a possible strategy we can use to take this gene, to clone it into the plasmid over here. All right, let's go through each answer choice one at a time. There might be more than one correct answer. Choice A says, let's use restriction enzyme KPN1 to cut both the plasmid, this thing over here, and the gene, this thing down here. And then you clone it. Well, let's look. KPN1, does it cut open the plasmid? Yes, right? It's right here. All right, now let's look at the gene. KPN1 cuts on the left, and it cuts on the right. That's very important. There might be a trick answer where they give you a restriction enzyme that only cuts on the left side, but doesn't cut the right side. That's useless. You can't use that one. Okay? So KPN1, because it cuts both the left and the right, and it cuts the plasmid, you can use KPN1 to insert this gene into the plasmid, because you create the puzzle pieces on the gene and on the plasmid so that they fit together. There might be more than one correct answer, though. Let's take a look. I'm actually going to skip B for a second because it takes a bit more to explain. So I'll talk about C first. This one says you're going to use restriction enzymes EcoR1 and KPN1 to cut the gene and the plasmid. So this time you're going to use 2 restriction enzymes. Well, that might still be fine. Let's go look at the plasmid. Is EcoR1 on the plasmid somewhere? Yep. Is KPN1 on the plasmid somewhere? Yep, right here also. Let's look on the gene here. Here's EcoR1, and here's KPN1. Note that there are 2 KPN1 sites on the gene, right? Doesn't matter that there's 2 KPN1 sites on the gene. Where's the EcoR1 site? It's also on the left. Just like as long as you have one thing on the left and one thing on the right, you are fine. So, because EcoR1 matches to the left and KPN1 matches to the right on the gene, you've created 2 puzzle piece ends. As long as those same 2 puzzle piece ends are in the gene, you're good. There's EcoR1 there, and there's KPN1 right there. So you are able to insert this gene into the plasmid using 2 restriction enzymes if you wanted to. I'll explain why sometimes you want to use 2 restriction enzymes instead of 1, and I'll explain that at the end. Well, if we were using test-taking strategies, because both A is true and C is true, that means all of them should work, right? Well, let's look at B just to make sure. It looks like there's 3 restriction enzymes here. We're going to try to use EcoR1 and XHOL to cut the gene. Okay, let's check to see if EcoR1 and XHO are here. EcoR1 is on the left, that's good, and XHO is there on the right, that's good. But then, for some reason, they decide that we need to use EcoR1 and SAL1 to cut the plasmid. Well, first let's see if they're even in there. Yep, EcoR1 is there and SAL1 is there. So we've opened up the plasmid correctly, and we've opened up the gene correctly, but normally, because we're using 2 different restriction enzymes for the gene versus the plasmid, that would normally not work, because you are creating 2 different puzzle piece ends. XHO shouldn't match with SAL1, and most of the time, if you get 2 different restriction enzymes, they won't match together. However, we're going to add one slight extra fact here. Let's look at how XHO and SAL actually cut the DNA that they're supposed to target. Like I said with EcoR1, EcoR1's job is to look for a specific pattern, and every time it finds that pattern, it's going to cut at that spot. That's true for any restriction enzyme. What is given here is the exact pattern that each restriction enzyme looks for. So we're only concerning ourselves with SAL and XHO, because we know EcoR1 works. That one is both in the gene and in the plasmid. But let's look at SAL1. SAL1 specifically looks for, and I'll zoom in because it's very important, SAL1 specifically looks for this pattern, G, T, C, G, A, C, and then it's going to cut at that arrow spot and that arrow spot. So almost at the end of this pattern, it's going to cut at each spot. And it creates an overhanging sequence, TCGA. The overhanging sequence here, and I would draw this here, but that's the sticky end. The areas between the arrows are your sticky ends. That's the actual puzzle pieces between the arrows. Okay, so it creates TCGA on one strand and AGCT on the other strand that are waiting to match with a corresponding puzzle piece. Let's look at XHO now. XHO, if you look at the whole sequence, it is technically a different pattern, right? Instead of GTCGAC, it is CTCGAG. However, look specifically at just between the arrows, and what do you notice? If you look at between the arrows for XHO, which is the only important part here, they have the exact same pattern, TCGA. Same as up here, TCGA. Let's check the bottom strand, AGCT, AGCT. If you only look at between the arrows, that is the sticky ends part. So even though this is a very unique situation, even though you are using two different restriction enzymes, because the sticky end pattern is the same, they will still fit together. So for the purposes of this question, you can treat XHO1 and SAL1 as the same enzyme. So now let's reread the answer choice and make sure it still makes sense. Now knowing that XHO and SAL are basically the same thing, we need to find an EcoR1 and XHO site on the gene. Check. Oh wait, on the gene. EcoR1 on the left, XHO on the right. Good. How about EcoR1 and SAL1 on the plasmid? EcoR1 here, SAL1 on the plasmid. We assume XHO and SAL are the same thing, therefore you can insert the gene into the plasmid like so. So all three choices will technically work to insert the gene into the plasmid. So that is one possible question they may ask, is which one of these can technically work? Another version of this question is which one is probably the best one to use. And the reason why I say the best one is more than it's just subjective, it's the following. So after you internalize that, after you go and practice this question, the next thing is which one is actually the best one to use? Well, let's look at choice A. Choice A, we're going to use KPN1 and that's it. So that means it's going to create a KPN1 site on the left and a KPN1 site on the right, and it's going to open up the plasmid. Whenever you use one restriction enzyme, you run into the following problem. When you use the same restriction enzyme on both sides of the gene, there is no way for the plasmid to tell which side of the gene is the front side and which side is the back side. So sometimes, when this gene is inserted into the plasmid, it's put in backwards due to